Well, let's get going on chapter 3. This chapter focuses on the molecules of life, essentially the title of this chapter. And the molecules of life are organic molecules. And the reason why we are referring to organic molecules as the molecules of life is because non-life, from what we can tell on Earth and our surrounding solar system, does not have these complex molecules. So they are unique to living things. Very big, complex molecules that have carbon as their foundation. Carbon is the element that forms the hub of these large organic molecules. Now, when I'm talking about organic molecules that are unique to life, I'm talking about things like proteins that you've heard about, carbohydrates, sugars, nucleic acids, which DNA is an example of that, and lipids, such as fats and oils, and steroids. So these are the molecules of life, and there are many, many different varieties of them. But as I was saying, carbon is the element that forms the hub of these molecules. Now you might remember, you should remember from chapter 2, that carbon atoms need 4 electrons to fill their second shell. So they need they need to make 4 bonds, 4 covalent bonds to get those electrons. So this allows multiple atoms to be attaching to carbons and making big complex molecules. You'll see examples of this as we go on in this chapter. The figure that's on the slide shows a scientist holding up a test tube with a pink band inside of it in the solution, and that pink band is DNA. We can isolate DNA from any living thing. We can sequence the information in that DNA and find out what the genes are, the instructions for building organisms. DNA is the information molecule that we have in every cell, and DNA has the information for making the other kinds of organic molecules that I've mentioned: proteins, carbohydrates, and lipids. So with this as an introduction, let's start exploring the molecules of life. If you were to examine a nutritional label on a food product, packaging, and by law, all packages containing food need to have nutritional labels on them listing certain information, the kind of information that in part you see in this figure. And you can see that there are organic molecules that are listed. They're required to the information for these molecules is required to be shared with consumers like you and me. So you can see fat is on there, and different kinds of fat, saturated fat, trans fat. We'll go over those. Cholesterol is listed on there. These are listed because these impact our health. That's why it's required for companies to tell us what's in these food products. Carbohydrates is listed. And remember I said on the last slide, carbohydrates are made up of sugars. And then finally, what's circled there in red is proteins. Those are listed there. So one kind of organic molecule that's not listed on nutritional labels is DNA. They're not required to tell you how much DNA is in your food. A nutritional label would extend down below that protein line, and if you look at a label, you would see that the various types of vitamins and minerals that are in your food would be listed below the protein. You can see iron is showing up at the bottom of that list. Now, there are tons of different kinds of organic molecules amongst the different life forms on Earth, a huge diversity, millions of different varieties, and these are made possible by clusters of atoms called functional groups. We're going to look at those on the next slide. Final note, as I say on the slide, functional groups give organic molecules their specific chemical properties. Functional groups in part determine how molecules chemically behave. And finally, the functional groups often are the parts of molecules that are participating or are involved in chemical reactions. So it's important for you as a biology student to become familiar with some of the basic functional groups, because you're learning about biology and you're going to learn about how some of these molecules interact and how they're important to us. So you need to know about these functional groups. Let's take a look at them. So there are 5 principal functional groups that you need to know about, because these are the super common ones that are found on organic molecules. And they're listed here in this table. What's most important for you to learn at this point from this table are the groups. So the groups include hydroxyl, carbonyl, carboxyl, amino, and phosphate. What's also shown in this table is the structural formula for each of these groups. And you need to be able to recognize those formulas or draw them. Indeed, drawing them would demonstrate to you or to me that you know what these functional groups are. So hydroxyl. Hydroxyl is an oxygen with a hydrogen attached to it. That's what it is. These are very abundant and common on organic molecules, and they are very important for determining the chemical properties. Carbonyl. Carbonyl has a carbon double bonded to an oxygen. That's it. That is a carbonyl. These are like faces, so you should be able to see these structures if I showed you a picture, like looking at someone's face, and go, aha, I recognize that face. That's a hydroxyl face. That's a carbonyl face, etc. Carboxyls, carbon double bond to an oxygen with an OH. Amino, nitrogen with 2 hydrogens attached. And finally, phosphate has a phosphorus with 4 oxygens surrounding it. Now, another important thing for you to understand from this slide and from those structural formulas is the line that's coming off of an atom in each functional group. So, for example, if you look at the hydroxyl on the top, there's a line coming off of the oxygen. If you look at the carbonyl, there are 2 lines coming off of the carbon. So what do those lines mean? What do they represent? Well, those represent the bonds that form between the functional group and the rest of the molecule. That's what they represent. So essentially, if you look at the hydroxyl, remember from chapter 2, oxygens need to make 2 bonds because they need 2 electrons. So there is a hydrogen already attached to that oxygen in the hydroxyl. The oxygen needs to make 1 more bond to be happy. So it does. It makes a bond with an atom in the rest of the molecule, and that's what that line represents. For the carbonyl, the carbons, of course, need to make 4 bonds to get their 4 electrons. Well, what the structure is showing you is the carbon is making 2 bonds with an oxygen, and then the carbon is making its other 2 bonds with other atoms inside of a molecule. That's what the line represents. Now, most organic molecules are really big. And for that reason, they are called macromolecules. Macro means big. So we put that prefix in front of molecule, and it's basically saying big molecule, macromolecules. So there are 4 categories of macromolecules in living things, and I've mentioned them to you already. So here's your chance to formally write them down and begin committing them to your memory. We are going to go over each of these types of or categories of macromolecules in this chapter. That's mostly what this chapter is about, is for you to get familiar with these different types. Proteins, nucleic acids, carbohydrates, and lipids. Let me say something briefly about each one of those, little bits of information to start associating with each one of those for you. Proteins. Proteins are the worker molecules of our cells. The cells of your body make tens of thousands of different kinds of proteins, doing specific jobs and functions to help your cells be alive, to support your metabolism, and to build and maintain different kinds of cells with different kinds of proteins. So when I say different kinds of cells, I mean like muscle cells, nerve cells, bone cells, etc. Nucleic acids. When you hear, read, see nucleic acids, you should be thinking genetic code. You should be thinking DNA or RNA. That's what should come to mind. Carbohydrates. When you see, hear, think carbohydrates, you should be thinking sugars. Carbohydrates include sugars. And finally, when you see, hear lipids, lipids are those remaining organic molecules that are non-polar, and they are hydrophobic. Okay, so these are terms from the last chapter. What kinds of molecules fall into the lipid group that are non-polar and hydrophobic? Well, fats and oils, they don't mix well with water because they're non-polar and hydrophobic. Steroids is another one. Waxes is another one. Think about it. You can't mix wax and water. Wax is hydrophobic. And another type called phospholipids. Really important. We will highlight each one of those. So that's what you should sort of think about at this point when you see or hear those different categories of macromolecules. Now, macromolecules are assembled or built from many similar small components or subunits. And these little components, molecular components or subunits, are called monomers. Monomer. Mono means one, and mero means part. This is Greek or Latin. So science uses Greek and Latin to make scientific terms. So think of monomers as little building blocks. You take little building blocks and put them together to make a bigger structure. You could think of monomers as being like little beads. You put beads together and you make like a necklace. And depending on which beads you string together, you make different kinds of necklaces. And then finally, the last point on here, if you take a bunch of monomers, if you take a bunch of little building blocks and put them together, you will make a polymer. So a polymer is a long chain of monomers. Look at the table that I have on the slide, table 3.1. So at the top there, one of the types of monomers is called amino acids. Those are little subunits, little building block molecules. And if you put together a whole bunch of amino acids into a long chain, you make a polypeptide, or what's being shown here in the table is protein. They're similar, but not exactly the same. I'll explain later. In the nucleic acid group, the monomers, the building blocks, are called nucleotides, etc. on this slide. Again, we're going to go over these different categories in more detail, but we're introducing the major categories of molecules on this slide, introducing the term monomer and polymer, and that's what you need to understand as you move on from this slide. All right, well, I just told you that monomers are the little subunits or building blocks, and polymers are the big long chains that form when you put those building blocks together. Now, polymers are all assembled or built in the same way. And that's what this slide is about, is showing you how polymers are made in cells. cells. This is happening inside cells. So look at the figure here. These pictures are super important. So on the top of the figure, you see a monomer on the right. It's like a sphere. It has an H and an OH. And then you see to the left of that a little chain of monomers, 2 of them. And what happens is off of one of the monomers, an OH, a hydroxyl is taken off, and off the other monomer, a hydrogen is taken off. You can essentially think of that as step 1. Step 1 in this chemical reaction. Hydroxyl removed and a hydrogen removed. Step 2 is those 2 parts linked together and form a water molecule. You can see that in the figure. And step 3 is the 2 monomers are then joined together, ka-chink, like linking 2 beads together on a string. Ka-chink, and now you have a slightly bigger molecule. You're building a polymer. And this kind of chemical reaction that's linking monomers by removing a molecule of water is called dehydration synthesis. So to dehydrate something means to take away water. So synthesis means to build or make something. So we are making water, dehydration synthesis. And in the process, these monomers, ka-chink, link together. Super important that you understand this, because this is the basis of how all the macromolecules, the polymers in your body are built. Your cells are doing this. So on the last slide, I explained to you how polymers are built by linking monomers together. How are polymers taken apart? How are they disassembled? It's essentially the reverse of dehydration synthesis. It's essentially the reverse. And this chemical reaction is called hydrolysis. Hydrolysis. Hydro means water, and lysis means to split or break up. So hydrolysis, hydrolysis means to split water. So what happens? Remember I said the steps are essentially the reverse of dehydration synthesis. So the first step here is a covalent bond between 2 monomers breaks. So if you look at the figure, you've got a chain of 3 monomers, 3 spheres, and the link between the one on the far right and the one in the middle is broken. Step 2. That water molecule is split into a hydroxyl and a hydrogen. That's step 2. And then step 3, this reverse process, is the hydroxyl attaches to one of the monomers and the hydrogen attaches to the other. And boom, finished. The polymer has successfully been split into smaller parts. This is hydrolysis. Where does this happen? Let me give you a quick example of when or where this is happening. This is how you digest your food. When you eat something, let's say you eat a sandwich, you are eating DNA, big polymers. You are eating proteins, big polymers. You are eating big carbohydrate molecules like starch. So you're eating polymers in your food. You need to split those apart into monomers. Number 1, so you can get them from your digestive system into your blood. And number 2, those monomers are what you use to build your own molecules, your human molecules, if you will. So digestion is an excellent example of when hydrolysis happens. So let me see here, losing my place. Here we go. So we're going to now survey these different kinds of organic molecules, these different categories, and we're going to begin with proteins. And in this PowerPoint, which is Chapter 3, Part 1, we're going to go through the protein category and we're going to stop there. And then in Part 2, we'll go over nucleic acids, carbohydrates, and lipids. So let's review the protein category. Proteins are complex macromolecules, very complex, very huge, and they are polymers. They are big long chains of little subunits, little amino acids, little monomers called amino acids. So the monomers of the protein group are amino acids. If you look at this figure, it's showing you this, it's summarizing this, it's showing you the structure of an amino acid, showing you molecularly what they are made up of, and we're going to go over that on the next slide. And then on the right hand side of this table, you can see a protein. And those little spheres are the amino acids, and they've been linked together by what kind of chemical reaction? A little review question. What kind of chemical reaction links the monomers together? Dehydration synthesis. So what you're seeing in that figure is an alanine, ALA, is bonded to a valine. The valine is bonded to a serine, etc. Different amino acids. So let's go on to the next slide and check out the structure of amino acids that you need to know about. Okay, so amino acids are the monomers of the protein category. They're not considered macromolecules. They're not big, huge molecules. They're little building blocks. And they have a pretty basic structure that you need to be familiar with. You need to be able to describe their structure, as I'm doing on this slide, be able to identify these different parts in a picture. Indeed, you should be able to draw your own picture of an amino acid. So let me go through this with you. Look closely at this figure as I'm going over this. So every amino acid has these components. A central carbon, which you can see in the figure as a C, that's kind of in the middle of the picture in the blue area. That central carbon has 4 things attached to it. 4 things. Remember, carbons need 4 bonds. So what you see in the picture is below the carbon is a hydrogen. Simple. It's got a hydrogen. Every amino acid has a hydrogen sticking off of that central carbon. If you look to the left of that central carbon, there is a functional group, and nitrogen with 2 hydrogens. Which functional group is that? We looked at it a few slides ago. That's the amino group. So every amino acid has an amino functional group. Now look to the right of that central carbon. There's a COOH. That's the carboxyl group, which you should recognize. That is on every amino acid. And then finally, looking up from that central carbon, there's an R with a circle around it. That's called the remainder group, or R group for short. And different amino acids have different remainder groups, clusters of atoms stuck together. In fact, there are 20 different amino acids that living things use to make proteins. So you and I have 20 different amino acids in the cells of our body. And these 20 different amino acids are made possible because each of the 20 has a different remainder group. So we're continuing with amino acids. Now, I don't expect you to learn all the 20 different amino acids. I don't expect you to recognize them or draw them. But what I do want you to know, and your book goes to this level, is that there are a few different categories of amino acids based on their chemical properties. So one of the categories of amino acids are the non-polar amino acids. And remember what that means. Non-polar means there's no partial electrical charges of negative and positive. These amino acids that are non-polar have remainder groups that are made up of carbon and hydrogen. Remember from chapter 2 that carbons and hydrogens share electrons equally enough because of their electronegativities that they form a non-polar covalent bond. So if you have a remainder group in an amino acid that's made up of carbons and hydrogens, that remainder group's going to be nonpolar. In the top right-hand corner, there's an example of a polar amino acid. So in the remainder group, that white boxed area, there are atoms that are not sharing electrons equally, and that makes the remainder group polar. So having a little bit of negative and a little bit of positive. So for example, the nitrogen that you see in the remainder group in that top right, it's connected to two hydrogens. Well, nitrogens and hydrogens don't share electrons equally. So that area of that remainder group would be polar. On the bottom left, there's an example of an ionized amino acid. So ionic, essentially. If you look in that white box, you can see the oxygen has a negative side next to it, negative sign. So that means it's ionized and it's got a full negative charge. There are 5 different amino acids that are ionized, either negative or positive. There are 6 that are polar, and there are 6 that are nonpolar. The remaining 3 are special kinds of amino acids that bond together with nearby ones in a protein and very much affect the shapes of the proteins. Okay, moving a step forward, talking about amino acids and proteins. When 2 amino acids bond together and make a larger molecule, the chemical bond between 2 amino acids is called a peptide bond. Peptide bond. And that is a bond that forms between an amino group in one amino acid and a carboxyl group in another amino acid. Look at the figure. These pictures and figures are always critical to help you see this stuff. So on the top of that picture, there's an amino acid on the right and one on the left. And it's showing that there's a hydrogen coming off of the amino group on the right, and an OH, a hydroxyl group, coming off of the amino acid on the left. That's step 1 of what kind of chemical reaction? Dehydration synthesis. The H and the OH bond together to make water. There's our dehydration. And then ka-chink, the 2 amino acids bond together, making a peptide bond, and we're starting to build a polypeptide. Polypeptide, many peptides. And if you look carefully at these pictures, that peptide bond is between the carbon that's in the carboxyl group on the left and between the nitrogen in the amino group on the right. This is what always happens 100% of the time when 2 amino acids bond together to start making a polypeptide. Okay, now that we reviewed what amino acids are and how amino acids bond together to make polypeptides, now let's start looking at protein structure. Protein structure is really complex. It's really complex because there's a lot of amino acids. Typically there's hundreds, if not thousands, of amino acids all bonded together to make one protein. And that molecule that forms, that big macromolecule protein, folds and twists into some kind of 3-dimensional shape that determines how it functions or works. Just like in your life that you experience, the 3-dimensional shapes of things determine how they function. You're holding a baseball, it's got a 3-dimensional shape, determines how it functions as a ball you can throw. You've got a baseball bat, it's got a 3-dimensional shape, long, and it can be used to hit the ball. So, just you could think of more and more examples of objects that have a particular 3-dimensional shape, and that shape determines how it functions. The same thing works in the world of molecules, and in this specific case, proteins. Ultimately, it's the order or the sequence of the amino acids in a polypeptide that determines how a protein is going to fold and twist together. I'm going to say that again because it's really important. The order or sequence of amino acids in a polypeptide determines how the resulting protein is going to fold and twist and function. Now, I'm showing you an example here on the slide, the protein called hemoglobin, which is found in your red blood cells. In fact, hemoglobin is what makes your blood look red. Hemoglobin is a globular protein, so think glob like a big blob. And hemoglobin consists of 4 polypeptides, 4 long chains of amino acids that are stuck together to make this protein we call hemoglobin. And hemoglobin's job or function is to carry oxygen in your blood. So the structure of hemoglobin determines its function, which is an oxygen carrier, an oxygen transporter. Now, we can give example after example of different proteins in your body that have different shapes and how those shapes determine how that protein works. Aha! Shape determines function. Now, as I've mentioned, different proteins have different functions, have different jobs, and what's listed on this slide is 5 functional categories of proteins. We can kind of group all the different proteins that your cells use into these 5 categories of what they do. There's actually a couple more categories that we'll probably see later on, but this is a good introductory beginning. Now, the first thing I'm noting on this slide is that proteins are called the worker molecules of cells. At least that's what I'm calling them because that's how I think about them. You literally make close to 100,000 different proteins in the cells of your body. And these proteins are what build your cells and make them work and do the jobs that they do, like muscle cells are for moving, for contraction. And nerve cells are for sending electrical signals through your body. Well, proteins are the molecules that are helping to build those different kinds of cells and make them do the jobs that they do. So the 5 categories that we're going over here include enzymes. You've almost certainly heard of enzymes before. Enzymes speed up chemical reactions, make them go faster. Almost every chemical reaction in your cells of your body has an enzyme to make it go faster. Transport, as the name implies, transport proteins help to move molecules from one place to another. Like hemoglobin in your red blood cells helps to transport oxygen. A third functional category listed here is structure. And there's 2 examples of structural proteins on this figure, on your slide. For example, in the X-ray of the human hand, it's pointing out that collagen is a structural protein that's found in your bones, your ligaments, etc., and helps to hold things together. In fact, almost 50% of the protein in your entire body is collagen, helping to hold things together. Another example of a structural protein on your slide is keratin, and keratin is what makes up hair. So there's a photograph of a lion with a bunch of hair coming off its head. That hair is made of keratin. So are our fingernails made of keratin. It's a very tough, strong structural protein. And the last one listed here, no, there's 2 more. Defense. So antibodies are defensive proteins. And so those antibodies stick to like bacteria and viruses that have invaded your body. And those defensive proteins called antibodies then trigger white blood cells to come and destroy those bacteria and viruses. So that's defensive. And finally, the last example given here is the contractile protein group. Contractile. When something contracts, that means it gets shorter. And that's what happens in your muscles, is that very long proteins contract, they get shorter, and that's what makes your muscles move. That's how you're able to move your fingers, that's how you're able to move your mouth or whatever, is contractile proteins, such as what's in muscle. So be familiar with these 5 functional categories and be able to briefly describe what each of them does, like I've done on this slide. Well, perhaps you've gotten the gist so far in this video, in this narration, that protein structure is really important to how they function, what jobs they do. And this is a primary focus in chemistry and biology, is to understand what gives proteins their structure. Because if we can understand how they get their structure, we understand how they work, we can use that information for medical purposes to help find cures and drugs and all kinds of things that are obviously very useful to us. So what we're going over now is a few slides to help you understand what gives a protein its structure or shape. There are 4 levels to protein structure, kind of 4 ways that the structure of a protein is built. And these 4 levels are simply called primary, that's the first level, secondary, that's the second, tertiary, it's the third, and quaternary means the fourth. The figure I put on this slide comes from the internet. You might have something similar in your book, and it is showing you these levels. Now I'm going to go through each of these levels of protein structure on the upcoming slides, but this is a summary on here. So primary level of structure focuses on the sequence of the amino acids in a polypeptide. That's the beginning, that's the starting point, is what is the order or sequence of amino acids in the big long chain. Now once you have a big long chain of amino acids, parts of that chain twist or coil, and parts of that chain can fold. Okay, kind of like the blinds in a window that are folding back and forth, or taking a piece of paper and like folding it to make pleats. So that's the secondary level, is the parts that are coiled into a helix and parts that are folded into what are called pleated sheets. The third level, the tertiary level, focuses on the 3-dimensional shape of a polypeptide when it twists and folds. And then finally, many proteins have a quaternary level. And the quaternary is the highest level of protein structure, and that's the overall shape that a protein has when there's 2 or more polypeptides that stick together to make the protein. So I've just given you a rundown of these 4 levels, but I'm going to give you a rundown slide by slide as we move forward here. Okay, so here is the primary level, primary structure. And here's a definition. I think I already told you what the primary level is on the last slide when I did a summary. But the primary level, you should be able to recognize this or even write it out on your own, is the exact order or sequence of amino acids in a polypeptide. That's what it is. The figure I put on the slide shows you a big, long polypeptide made up of over 125 amino acids, like little beads. So if a chemist or a biologist wanted to know the primary structure of this polypeptide that's called transthyretin, that chemist or biologist would literally be figuring out the order or sequence of amino acids. Now, each of these amino acids, like we reviewed a little bit ago, has a remainder group or R group that's sticking out. There's 20 different ones. So ultimately, the order or sequence of those remainder groups going down the chain is going to determine how that polypeptide is going to twist and fold into its 3-dimensional shape. So this is a very important starting point. Now, the order or sequence of amino acids, the primary level of structure, is determined by the information in your DNA. Like that's what much of your DNA is all about, is having information, genetic code, for which amino acids to grab in your cells and build proteins. So that's what determines the sequence or order of the amino acids, is your genetic code, your DNA. Okay, let's jump to the secondary level of protein structure. The secondary level, the definition of it, I put in blue, is the segments or pieces of a polypeptide that might be coiled into what's called an alpha helix, or folded into what's called a pleated sheet, a beta pleated sheet. And so what I've done is I've included a figure on the upper right-hand side of your slide, and it shows a twisted ribbon-like structure that is a polypeptide. And there's two parts in that polypeptide that are highlighted, that are blue. One part is coiled, that is showing you an alpha helix, and another part is folded, and that's showing you pleated sheets. So the way that these form is you can see, if you look really closely, is that the amino group in one amino acid is attracting to part of a carboxyl group in another one. And those attractions cause that polypeptide to either make coils or folds. So that's what's making these this secondary level of structure, is those attractions. And yeah, look, notice, like from chapter 2, those are little dots drawn in between those different parts of the polypeptide. And what do the dots represent? Hydrogen bonds. So hydrogen bonds are helping to hold these shapes together. Hydrogen bonds are helping to hold the coils and the folds in place. What's an example of a protein that has lots of folds in it and gives it a really strong structure? Spider silk. So I put a photograph of a spider on its spider web. Spider webs are made of spider silk that spiders produce using the DNA in their cells that has the genetic code or information for making the protein for silk. Believe it or not, spider silk is stronger than steel. Pretty remarkable. So if you have two thin strands, one is a thin strand of spider silk and the other is a thin strand of steel, the spider silk would be stronger. Spider webs are pretty strong, and it's made possible by all these pleated sheets in the spider silk that are making hydrogen bonds and holding those shapes together. Makes it really, really strong. So that's the secondary level. Coils and folds in a polypeptide. All right, well now we've come to the tertiary or third level of structure that builds a protein. The definition of the tertiary level of structure is the overall three-dimensional shape of a polypeptide. That's the definition. You should recognize that, be able to write it out. The overall three-dimensional shape of a polypeptide. And this overall three-dimensional shape is a result of the R groups or remainder groups interacting and forming different kinds of bonds, etc. And so this polypeptide, remember, is a long chain of amino acids, like a long necklace with a bunch of beads. And so what I'm saying is that long chain or long necklace is folding and twisting into a three-dimensional shape, and that is determined, that shape that it folds and twists into overall is caused by the remainder groups. You can see, I've put little asterisks here, some of the interactions that occur between remainder groups. For example, remainder groups that are hydrophobic, that are nonpolar and hydrophobic, they consist of carbons and hydrogens. They tend to huddle in the middle of a protein, hiding from the water that surrounds the protein in our cellular environment. Polar remainder groups form hydrogen bonds. Check out the figure I'm showing you here. So there's little remainder group sticking down. It's got a CH2 and an O, and a remainder group sticking up from another part of the polypeptide, and they are attracted to each other with a hydrogen bond. That part helps to make that twist. Other parts, other amino acids are attracted because they're ionic, and they form ionic bonds, so they're ionized. So a remainder group that's negatively charged, like the one shown in the figure, that's got an oxygen that's negative, will attract to another remainder group that's positively charged, like the one shown in the picture. And that'll help make the polypeptide twist in that particular direction. And then finally, another really important type of interaction that makes the polypeptide fold and twist is called a disulfide bridge. And that's when sulfur atoms that are in two different remainder groups form a covalent bond. And that is called a disulfide bridge. Di means two, sulfide refers to the sulfur. Remember, covalent bonds are the strongest kinds of bonds. So if a protein has disulfide bridges that are holding its three-dimensional shape, those are really strong interactions or bonds that are holding that shape. They're like nails that are being pounded into something to hold it in place and give it shape. That's what disulfide bridges help to do. All right, here's the fourth and final level of protein structure. Now, not all proteins have this level. Not all proteins go up to the quaternary level. Some stop at the tertiary level. A protein only has a quaternary level of structure if it's made up of two or more polypeptides that are stuck together. So, the definition of the quaternary level of structure is the overall shape of a protein when it has two or more polypeptides that are stuck together. That's the definition. So I'm showing you again a figure on the upper right-hand side of the slide of hemoglobin. There's hemoglobin again. And what I want to point out to you here is that hemoglobin is made up of 4 polypeptides. 2 of them are called alpha subunits. They're identical. And 2 of them are called beta subunits. They're identical, so they're essentially different kinds of polypeptides. And they all stick together in a very specific way. And when they do, we have the protein called hemoglobin. So this kind of brings us to the question, what is a protein? What is a protein? A protein is a biologically active molecule made of 1 or more polypeptides. Biologically active means it has a job or a function in a living thing. So some proteins are only made of 1 polypeptide that twists and folds into a 3-dimensional shape. So that would mean that that protein stops at the tertiary level if it just has 1 polypeptide. But like hemoglobin, if a protein has 2 or 3 or 4 polypeptides, then it now has a quaternary level as well. So I'm showing you hemoglobin here, but look at the bottom of the slide. I'm also showing you collagen. Look how collagen is made of 3 polypeptides that are twisted around each other like a rope. And remember, I mentioned this a little earlier, collagen is a structural protein. It's helping to hold things together in your body. So a rope-like structure seems like it would work well for structure. So collagen is rope-like. About 40% of our protein in the human body is collagen. We have a lot of it in our bodies. So wrapping up, this is the 4th level of protein structure, only if a protein has 2 or more polypeptides. And what is a protein? Went over that. And then 2 examples on here of proteins with a quaternary level of structure. Okay, well, we have now gotten to the point where we have reviewed what a protein is, what they're made up of, amino acids, polypeptides, that their shape determines their structure. So because of that, we went over in detail the different components that give a protein its structure. The last few slides, we went over that. Shape, super important to how things work. Now, in a normal watery environment of cells, proteins fold and twist into their functional shapes all by themselves. Well, kind of all by themselves. We'll come back to that in the next slide. There are certain environmental factors, variables, that can cause a protein to change its shape and not work anymore. And when a protein changes its shape and doesn't work anymore, then it has denatured proteins. There are 3 environmental factors that very commonly can cause a protein to denature: high temperatures, high salinity, salt, or changes in pH. So low temperatures don't cause proteins to denature. In fact, low temperatures help to stabilize protein shape. But high temperature, remember what high temperature is. It means molecules are moving really fast with a lot of kinetic energy. So if there's a lot of kinetic energy going on at high temperature, it could cause the bonds to break that are holding a protein together, and it loses its shape. Remember, salt is made up of a chloride ion and a sodium ion, and with strong electrical charges. So if certain proteins are surrounded by a lot of those ions, there's high salinity, high salt, those charges of the ions could cause the protein to unravel or denature. And finally, remember what pH is. It's a measure of the concentration of hydrogen ions and hydroxide ions. We covered that in the last chapter. So proteins have a normal shape at a specific place in the pH scale, usually in living things close to pH 7. And if that pH shifts and becomes more acidic or more basic, it could cause a protein to denature because those ions are very reactive. So I have an example on here with egg white. Most people have seen or cooked an egg. And when you crack that egg open into a hot frying pan, the egg white is made of a protein called albumin. That's the protein. It's very important to eggs. And when that albumin hits the hot surface of a frying pan, high temperature, it causes the structure of the albumin to denature, to change shape, and it kind of relinks into different shapes, and you get that gelatinous egg white. And you can't go back, right? Once you cook an egg white and it becomes like gelatinous, it doesn't go back to its normal shape. So many times denatured proteins stay denatured, and that would be deadly. So this is kind of a topic to point out. Denaturing is not good for living things. If a living thing, whether it's a bacterium or a plant or a human, has its proteins denaturing because of high temperatures or high salinity or pH changes, that could be deadly. Last comment about this. Think about like high fevers. You get really sick. A high fever could kill you. If you get a fever of 105 degrees for too long, be dead. What's happening? Well, a big part of what's happening is certain proteins in your body are beginning to denature, and that'll kill you. I've mentioned it many times in this PowerPoint, this part 1 of chapter 3, that protein structure determines function. And this is the last ending slide in this part 1 that is ending on this topic. Protein structure determines function. So remember the phenomenon of what we called emergent properties in chapter 1, that when components or parts are put together in a specific way into a bigger thing, a bigger system, that bigger thing now has emergent properties, has new characteristics that didn't exist before. Like the emergent properties of your hand, being able to grasp and manipulate things because of the way all the components in your hand are put together. Or a computer or a car functioning because all the components are put together in a certain way, so you get emergent properties. Well, the same thing applies in the world of molecules. When a bunch of monomers are put together to form polymers and they have a three-dimensional shape, three-dimensional structure, that three-dimensional shape or structure determines how those molecules are going to function. So one important example here is that category, a group of proteins called enzymes. Enzymes have the job of making chemical reactions go faster. And enzymes work by having a little pocket, a little space inside of the enzyme molecule called an active site. That's what the little space or compartment is called. And very specific chemical reactants fit into that active site, into that little cavity. And that shape of the active site, that shape of that cavity is what makes that enzyme work in its specific way. Shape determines function. We'll come back to enzymes in a future chapter because you understanding what they do and how they work is one of those things in Bio 156, in introductory biology, that is crucial, especially if you're going to move on and take, say, microbiology or physiology. You've got to understand enzymes and how they work. So we will come back to that. So the final thing here is, well, some proteins need help in getting their final functional shape. And there are certain proteins in your body that help other proteins get their correct shape. And those proteins are called chaperone proteins. And there's a figure on the bottom of your slide that's showing you how they work. So there's a little compartment inside of a chaperone protein, and a misfolded protein, a protein that doesn't have the right shape, fits into that compartment. A little cap goes on the top, and the environment in that chaperone protein allows the misfolded one to fold correctly. The cap comes off, the properly folded protein pops out, and then it can do its job. So chaperone proteins are really important for some of the proteins and cells to be able to get their correct shape, because otherwise the cellular environment wouldn't allow them to. So this wraps up our protein category. There's lots of stuff for you to understand about proteins. And as I said, in part 2, that'll come a little bit later, I will review the other kinds of organic molecules, nucleic acids, carbohydrates, and lipids.